Alright, so we're going to start with a review of what we've covered so far. And we started with chapter 7, which is respiration. Again, because this is the A2 section of the course and following the pattern of the Cambridge syllabus. So, respiration is the chemical breakdown of substances to release energy, right? And so, glucose is used and ATP is produced. So, the energy released from the breakdown of glucose is stored as ATP molecules, and right when the cell requires energy, those ATP molecules are utilized to provide the energy. Right? And why do we need energy? Because of everything that's going on in the body. So transport, building of molecules, DNA replication, cell division, and even at rest, right? There's a lot going on. And then we have the brain, obviously, and the nervous system, which is the highest consumer of energy in the body, right? Contributing to only 2% of the body weight, but up to 20% any energy consumption. Then understanding the difference between respiration and breathing. So respiration is a chemical process and it occurs in all the cells of the body, in the cytoplasm and the mitochondria. Whereas breathing is the physical process of inhalation and exhalation of air, right, where gaseous exchange occurs in the lungs. The overall equation for respiration is given over here, where you have a molecule of glucose that combines with oxygen, and then you have carbon dioxide given off as a base product, water is produced, and ATP, right? And that is the energy produced. The ATP molecule, right, it's adenosine triphosphate. So it is the adenine nucleotide along with 3 phosphate groups. And it is, right, a reversible reaction where the ATP can be hydrolyzed to remove one of the phosphates and release 30.5 kilojoules of energy. This is reversible because the phosphate can be rejoined, right, through phosphorylation of the ADP to get back ATP. Okay. Now, ATP cannot be stored in large quantities. There is a small reserve, but whenever energy is required, the machinery to make ATP is present in the cells, right? The initial stages of respiration take place in the cytoplasm, and then everything else in the mitochondria. It is a multi-step process which includes, right, starts with glycolysis, and then there's the Krebs cycle. There's also something called the link reaction in between, and all of this, right, culminates in the electron transport chain, right? And that is the part where oxygen is actually utilized and the bulk of the ATP is produced. And we're talking about aerobic respiration, so that's happening in the presence of oxygen. We came across, in fact, right, hydrogen acceptors. So we've got NAD, which is nucleamide adenine dinucleotide, and then FAD, flavin adenine dinucleotide. Both of these are coenzymes, so they assist enzymes or small molecules, assist in enzyme catalyzed reactions. And when hydrogen is removed in the process of glycolysis, they're picked up by either NAD mostly or FAD, and these become reduced NAD and reduced FAD, and they move on to the last part of the process, which is the electron transport chain. Right, to keep in mind that oxidation occurs when an electron is lost, oxygen is gained, or hydrogen is lost, and similarly reduction occurs when an electron is gained, oxygen is lost, or hydrogen is gained. Then, right, we had already discussed the structure of the mitochondria when we were talking about organelles in the AS classes. All right, so just one thing to highlight, that the inner mitochondrial membrane, the cristae, have these stalk particles which are ATPase molecules. So they're enzymes that catalyze the conversion of ADP and inorganic phosphate to ATP. The process of glycolysis is where a 6-carbon glucose molecule is converted to 2 molecules of 3-carbon pyruvate. Right? And this is how it is carried out. So glucose is converted to, right, a phosphorylated 6-carbon sugar, and then that is broken into two molecules of a 3-carbon sugar, and then that is converted to pyruvate. Initially, ATP is utilized, but then later on it is produced, and right, you also get reduced NAD. Then moving on to the Krebs cycle, before that, right, there is what is called link reaction. Right, so the glycolysis takes place in the cytoplasm of the cell, and everything else, the link reaction, the Krebs cycle, the oxidative phosphorylation or the electron transport chain, it occurs in the matrix. Well, the link reaction and the Krebs cycle occur in the matrix of the mitochondria, and the electron transport chain and oxidative phosphorylation occur at the inner mitochondrial membrane, the crystal. So in the link reaction, you have a 3-carbon pyruvate that is converted to a 2-carbon acetyl coenzyme A. Right? The overall reaction that occurs is the pyruvate combines with coenzyme A, and NAD is also involved. It's converted to acetyl coenzyme A, which is a 2-carbon molecule. Carbon dioxide is released, so that's the first release of the waste carbon dioxide, and you get reduced NAD. So the pyruvate passes into the mitochondria, and right, a molecule of carbon dioxide is removed from the pyruvate, so from the 3-carbon molecule. That is called decarboxylation. A 2-carbon compound is formed, which is combined with coenzyme A to form acetyl coenzyme. Right, pyruvate is also oxidized, so hydrogen is removed, which is picked up by NAD to form reduced NAD. And the decarboxylation is carried out by enzymes called decarboxylases, and the removal of the hydrogen is carried out by enzymes called dehydrogenase. Then we come to the Krebs cycle. Over here we have the 2-carbon acetyl coenzyme A that combines, right, so coming in from the link reaction, and we have a 4-carbon molecule, right, which is called oxaloacetate, so that's not mentioned in the Edexcel book, and you end up with a 6-carbon molecule, which is citrate or citric acid, and the coenzyme A, which was used in the link reaction, right, so over here, that is regenerated, and that goes back to take part in the link reaction further. Now, what happens is that the 6-carbon, right, this citrate enters a cyclical process where 2 carbon atoms are removed. So, right, 2 molecules of CO2 are produced, 3 molecules of NAD are reduced, right? That means 3 hydrogens are removed. 1 molecule of FAD is also reduced, so that adds 1 more, so that's 4 molecules of hydrogen, and 1 ATP is produced, right? So again, these steps are not the not where the bulk of the ATP is produced. And after the entire process, you get back, right, the 4-carbon compound that you started off with, which combined with the acetyl coenzyme A coming in from the link reaction. And so that's why it is a cyclical process. That's why it's called the Krebs cycle. And right, this is happening twice, because you started off with 1 molecule of glucose, which became 2 molecules of pyruvate, which gave you 2 molecules of acetyl coenzyme A, and each of those molecules will undergo the Krebs cycle once, and so the Krebs cycle is happening twice for a single molecule of glucose. Then we have the electron transport chain, right, and oxidative phosphorylation. So the link reaction and the Krebs cycle, as I mentioned, occur in the matrix of the mitochondria, and the electron transport chain and oxidative phosphorylation occur at the inner mitochondrial membrane, because it has proteins, which are electron carriers, and the stop particles mentioned earlier, which are the ATP synthase or ATPase molecules. Now what happens over here is the reduced NAD and reduced FAD come in, and they give up the hydrogen that they picked up, and the hydrogen is broken up as a proton and an electron, right? And then the NAD and FAD are oxidized again back to the way they were, and those go back to be reused in the process of respiration. Now the electron from the hydrogen, right, because hydrogen is basically a proton and an electron, that's all it is. So the electron from the hydrogen is passed along a series of electron carriers, so those are proteins, in a series of redox reactions. So when one of the proteins takes up the electron, it becomes reduced, and when it passes it on, so it loses the electron, it becomes oxidized, and the next carrier becomes reduced. And so, right, when both of those are happening, right, it's called a redox reaction, which basically means that reduction and oxidation both are occurring as part of the process. Okay. So I mentioned that, and there are about 4 to 5 steps, right, that occur. There are different electron acceptors, electron carriers, and at the end of the process, right, there is the oxygen, right? So that's where oxygen comes in, the one we got from breathing, right? The gaseous exchange in the lungs at the alveoli. And oxygen is what ends up accepting the electrons as well as the protons, right? So all of this, right, the journey that started from glycolysis, right, the hydrogen, right, broken up as the proton and electron, is combined with oxygen to form water. Okay, so the carbon dioxide that we get when we exhale, right, as the waste product, comes from the molecules, the breakdown of glucose into, right, simpler molecules, so to speak. And the oxygen is actually, right, so it's not used to make carbon dioxide, but it is used to, right, react with the hydrogen and proton and the electron to make water. So this water, right, it might be utilized in the body, right, or excreted, or whatever the end result of the water is. Now the proton, right, there's something else going on over here. So the electron is passed along the electron carriers in the electron transport chain, and as it passes along, it loses energy, which makes sense, right? But the energy of the electron is used to actively pump protons into the intermembrane space of the mitochondria, right? So when it says actively, we're talking about active transport. And active transport is a process where something is moved from lower concentration to higher concentration against the concentration gradient. It requires energy. Usually that energy is provided by ATP, but in this case, it is provided by the electron, right? And so there is a very high concentration of protons in the intermembrane space, which makes it acidic, right? And so the protons want to move back into the matrix of the mitochondria. There is a concentration gradient, there is a pH gradient, and there is an electrochemical gradient, right? So too much of the positive ions in the intermembrane space, right? So, right, there's a high concentration of positive ions, and they want to move back into the matrix. So that's the electrochemical gradient, along with the concentration gradient, right? Because there are a lot of protons there, and the pH gradient, right, should become acidic, and so they want to move back into the matrix, which is less acidic, right? And so what happens is that the protons move back into the matrix, but right through the ATP synthase molecule, the stock particles, right? And that is where this reaction takes place, right? And along with that, the energy of the the passive diffusion, right, or just the diffusion of protons through the ATP synthase molecule. So there's energy that, right, there as well, and so that energy is used to synthesize ATP, and that is where the bulk of the ATP comes in. And right, this entire process is referred to as the chemiosmotic theory. Finally, how much ATP is made, right, or produced from one molecule of glucose? So different values are given, right? Probably different experimental conditions or different conditions of the body. And right, 38, 36, 31, those are all figures, but right, there is a consensus, I guess, or a safe figure that the number of ATP molecules produced from one molecule of glucose is rarely above 30. All right. So that is a review. Any questions?
No, sir.
All right. Now moving on. Okay. So now we're going to talk about anaerobic respiration. So that is right, chapter 7A/2, right? And this is not the heading of the chapter, right? The topic of anaerobic respiration is covered in this chapter, right? And as the name suggests, we know aerobic, right, means the presence of O2, and an means no, right? So the respiration that takes place when there is no oxygen present, right? And right, there are conditions where, right, when cells to respire, right, without, or right, when there's a lack, lack means there's not sufficient, right, lack of oxygen. An example of that scenario, right, for humans at least, would be the muscle cells, right, during physical activity, and not just demanding physical activity, which means you're exercising in some way, playing a sport or jogging or doing something. And so why, right, why is anaerobic respiration important, and why it actually occurs in the cells, right, is because the organism, right, the systems of the organism, right, are programmed to survive. So it's not like there's going to be a lack of oxygen and they're just going to say, oh, all right, so we tried and there's no more oxygen, so I'll see you later. It's not how it is. And so you could say that anaerobic respiration is sort of a fail-safe, right, and a protective mechanism to keep the organism going. Just a moment. So, as mentioned in the case of exercise, right, oxygen is not supplied fast enough to meet the energy requirement of the muscles, right? So whatever oxygen they had, they utilized it for the ATP, and the ATP is all utilized for muscle contraction, right? That you're exercising, and it's still not enough, right? So when you exercise, your breathing rate goes up, you start breathing deeper and faster, you start taking more air, but there's a limit to how much you can do, and right, and it's just not enough. And so to provide ATP to keep the muscles going, right, they utilized anaerobic respiration. Now keep in mind that the ATP output is very low. It's very low, right? So compared to the the figure that we had for aerobic respiration, we just go back to that. Right, we had 2,880 kilojoules for aerobic respiration. The output of ATP, right, the energy is just 150 kilojoules. And it's less than 10%. Okay, now going into detail, right? How does this happen? So in the muscle cells, right, glycolysis does occur. you have glycolysis occurring, and right, you get pyruvate at the end. Right, but that pyruvate cannot move. Right, this does not move. You can say does not move into the mitochondria. Right, due to lack of oxygen, and so it takes a different path where the pyruvate is converted into a compound called lactate. Okay, and along with that, right, you have the release of energy, right? So you've got some ATP being produced, and right, the what happens is that right, the pyruvate sort of goes into a cyclical process. So right, NAD is involved. Right, so pyruvate is a 3-carbon molecule, and this is also a 3-carbon molecule. But what's happening over here is hydrogen is being lost. Sorry, my bad. Okay, this is. Can I just fix this? It's still. My pen is glitching. There we go. Type that out. It's still glitching. All right, so my whiteboard glitched, so sorry about that. I got a bit distracted. So, I I said a hydrogen is lost. It's actually gained. So sorry about the confusion. Right, and so pyruvate is reduced. Right, and what's reducing it is the reduced NAD that was gained from glycolysis. Okay, so that reduced NAD gives up its hydrogen, so that's the one giving up the hydrogen, right, and going back to becoming NAD, and then going back into the process of glycolysis, right, and so that's how NAD maintains, you know, both oxidizes and is maintained. So the process of glycolysis is continuing, and right from there you got reduced NAD, and then the reduced NAD converts the pyruvate, it reduces the pyruvate to lactate. Okay, and then the NAD goes back into glycolysis. Now the lactate is accumulated. Okay, so the lactate is, as long as there's the lack of oxygen and oxygen debt, right, the lactate builds up. And the only source of ATP is glycolysis, the first step. And there are only 2 molecules of ATP being produced, right, via glycolysis, which is obviously very low, right, as compared to 8 molecules of ATP produced via glycolysis under aerobic conditions, right, in the present. So, right, for just for this step, you've only got 25% product, efficiency for the number of ATP molecules being produced, and the rest of the cycle, the process isn't even happening. Now, what happens later, right, when you stop exercising, there's an oxygen debt. So when you stop exercising or whatever it is that you're doing that created the oxygen debt, right? You take deep breaths for some time, and this continues. It could be 10 minutes, 15, half an hour, depending on how severe it was and overall body health, right? And so you're actually taking in a bit of extra oxygen for some time than you would, right, compared to what you would normally. And what happens is why you need that extra oxygen is because the lactate is moved to the liver, right? And the oxygen is utilized to oxidize it and then utilize it in glycolysis, right? So it is reconverted back to pyruvate, and then, right, it is fed back into the process of respiration. So it doesn't go to waste, right? Pyruvate is reformed due to the oxidation of lactate, and then the pyruvate is fed into the link reaction and Krebs cycle and so on. And because oxygen is now present, the rest of the process of respiration can occur. Okay, now, right, you experience muscle fatigue, right? Your muscles seem to lose power, let's say, strength, especially under when you are performing vigorous exercise and, right, you're experiencing this anaerobic condition. And right, it used to be believed that it was because of the buildup of lactate in the muscles, but now it is believed that the lactate, right, and, right, hydrogen ions, they enter the blood, and that lowers the pH and the high lactate concentration in the blood, right? When that goes to the brain, right, it affects the nervous system. And that affects muscle contraction. So it's a indirect effect. And so the muscle contraction reduces, right? Because muscle contraction also occurs due to nerve impulses coming in, right? So the nervous system is affected by the pH and the lactate buildup. So, right, the muscle contraction will obviously be affected as well. Now, right, this was an example. Any questions?
No, sir.
Okay, sorry, it was a bit.
No, sir. It's okay.
Okay. Now, anaerobic respiration also occurs in other organisms, right? So you have got yeast, for example. Right, now organisms obviously prefer aerobic conditions, right? But they do experience anaerobic conditions. And as I mentioned, right, the anaerobic respiration is a mechanism to keep the organism alive. So, right, in plants, right, so that occurs in plants as well. An example is rice, right? So rice fields are flooded with water, and so the roots don't get oxygen because obviously the oxygen concentration in the soil is low compared to the air anyway, and then it's submerged in water, and so, right, that further reduces the oxygen available to the roots, and so that creates anaerobic conditions, right? So anaerobic respiration occurs in plants as well. Then you've got, right, these the yeast organism, And what happens here is slightly different, right? So not going into detail, but just giving you, right, the overall equation. So you've got glucose, and that is converted to ethanol. And carbon dioxide is released, right? And some ATP is produced, and you can see that the energy that is gained is even lower than what we had in the muscles, right? About 144 kilojoules. If you want the chemical equation, you've got C6H12O6, right, without oxygen, and you are getting two molecules of ethanol because it's a two carbon compound. C2H5OH along with 2CO2 and ATP, right, and the same energy. And the thing about ethanol is, right, unlike lactate, which is converted back to pyruvate and then reused, right, ethanol does not go back to being glucose, right? Acha, glucose obviously, right, to be clear, right, glycolysis occurring, so glucose goes to pyruvate, and that goes to ethanol, just to be clear, right? It's not like glucose is being directly converted into ethanol. So, right, but the ethanol cannot go back to being pyruvate, and so it builds up. And if the yeast is kept in anaerobic conditions for long enough, it will die, not because of the lack of oxygen, but because of the ethanol, which is toxic. But the fact that it can produce ethanol under anaerobic conditions is actually utilized in our industries and even at home, right? So when you add yeast to bread, to dough, actually, not bread, when you add yeast to dough, when, right, you put flour and water and you mix it together and you put a bit of yeast and you leave it, and you see after half an hour or something, the dough rises. And that rising of the dough is because, right, the yeast that you've added is undergoing anaerobic respiration in the dough, and it's rising because the carbon dioxide is being produced, right? And then you put it in the oven, and the ethanol is also produced, but, right, it evaporates due to the heat. And when you eat bread, you might have seen these sort of holes, right? And that is where the carbon dioxide bubbles were, and because of the heat, they're gone, and so they've left those spaces in the bread. Okay, it's also used. This yeast fermentation is also used to produce alcohol industrially, right? So it has its benefits for us. Any questions?
No.
Okay. Another thing I left out was, yes, investigating the factors affecting the rate of respiration. Right? So that is an experimental setup that is mentioned in 7A1. And, right, for this one, I'll actually move to the book. It will be easier to explain because they have an illustration. So just give me a sec. Can you see the book?
Yes, sir.
Okay. So as they say over here, right, what we're talking about is not a sophisticated setup where they're measuring respiration organelles, but a school setup where something called a respirometer can be used. And what they're going to use, right, is some small organism that is respiring, right? So they're going to measure the uptake of oxygen or the output of carbon dioxide by whole organisms, right? And that whole organism can be a small insect or germinating seeds, whatever is respiring. So a basic respirometer consists of a closed chamber with in which nowhere air can enter, right? And contains one or more living organisms. So there are two different setups for this. This is a more basic setup, as you can see on the left, right? There is a tube, right, with the seeds apparently over here, and right, so those are respiring. There's air inside, so there's obviously oxygen as well, and the carbon dioxide that is released can be taken up by either using soda lime or potassium hydroxide, and so that will absorb the carbon dioxide. And the good that will do is, right, so if oxygen is taken in, carbon dioxide is released, the volumes are almost the same, and so, right, we won't get to know how much oxygen was utilized, right? And the way that we're going to measure this is there's a liquid in the manometer over here, and as oxygen is utilized, the pressure of air in the tube that is pushing on the liquid in the manometer will decrease, and so the level of this liquid will rise, right? So if oxygen is taken up and carbon dioxide is released, and so the volume of air over here when the tube remains constant, and so you will not observe any change, and that's why you have the soda lime to absorb the carbon dioxide, right? The gauze is so that the seeds are separated from the soda lime. They don't fall on the seeds and cause problems. Okay. So as the organism uses oxygen, the pressure reduces, and so the fluid in the manometer moves upwards towards the tube, right, containing the organisms, and right, a syringe, right, can be used to measure the volume of gas need to return to the manometer to normal, right? So this is a syringe over here, and so you made a note of where the level of the liquid was originally, and then it rose, and so there are no markings on this tube, right, the manometer. And so when you add some air back into the tube, right, so we're talking about volume over here, so volume of whatever gas, it doesn't really matter, it's a volume, right? And we know that gases take up the volume that is available, right, the space that is available. And so we add air back into the tube, so that's going to increase the pressure again, and that will move the liquid back down to the level where you started. And then you measure how much air you put back in, and that will give you, right, how much oxygen was utilized, right? And so you can use this measurement to calculate the intake of oxygen per minute, right? So how long did you leave this experiment to run? Did you leave it for 5 minutes, 10 minutes, half an hour, an hour? It depends, right? And so, for example, you, right, to keep the calculation simple, you left it for 10 minutes, and then you put the air back in, and you notice that 10 centimeter cube of air was required to return the level of the liquid in the manometer back to the starting point, right? And so you measure the rate of intake of oxygen. So 10 centimeter cube of oxygen was utilized by the seeds in 10 minutes. So that's 1 centimeter cube of oxygen per minute, right? And that is an approximation, obviously, right? Because it's not a very sophisticated setup. And then what we can do is that we can change the external conditions. So you can see that a water bath is used to keep the temperature constant. We can change the temperature, for example, and see how that affects the rate of respiration, right? So increase in temperature, what effect that would have on the uptake of oxygen. Okay. And there are obviously limitations, right? But it can give you an overall idea about it. Okay. Then, right, there's another investigation investigating the site of ATP synthesis and how that takes place. Right, you can investigate respiration at the cellular level in a number of ways. You can break open cells and centrifuge the contents to obtain fractions containing just mitochondria. Right, so this is just like a summarized outline of how that will happen. Right, not details of right what the sedimentation rate of mitochondria are, which layer it will be in, etc., etc. Right, how to break up the cells, how to treat them. And then if these mitochondria are supplied with glucose and oxygen, they will produce ATP, they'll do their job. And using higher resolving electron microscope, so obviously this is not something you'll do in your school lab. You can see that the surface of the inner membrane of the mitochondria is covered with closely packed stock particles, and these provide a greatly increased surface area, which is the ideal site for enzymes to work. And then what you can do is you can separate the stock particles and the small pieces of membrane associated with them from the rest of the mitochondrial structures. And scientists have demonstrated that ATP synthesis only occurs here. This and other evidence proves that the stock particles are vital for the formation of ATP. Right? So just two experimental procedures that were given about the rate of respiration and the site of ATP synthesis. The next time we will move on chapter, just a minute, 7A5, I believe. Yes. So the next time we'll be moving on to chapter 7A5, where we'll be talking about respiratory substrates. Any questions?
No.
All right, so do go through the content in the book,
Yes.
right? And if there's anything, obviously feel free to ask. And I'll see you tomorrow, and we'll continue forward from here.
Yes, sir.
All right?
Yes.
All right. Thank you.
Reference.
Allah